brought to my attention something in the book that may cause some confusion. When you're studying interferon, there are 2 pages that the index sends you to, 442 and 447. And 447 gives you a little bit more detail about interferon. Interferon is huge, and there's all kinds of research on interferon and all the things it does and how it interacts with the immune system and how it works when you give it exogenously and all that kind of stuff. When you read 447, it's not as clear as I taught in terms of what alpha, beta, and gamma do. It gives you the impression, and it's not a false impression, but it's just more detail than you need. It lets you know that alpha and beta also have some activity on phagocytes. But the big picture, and that's the picture I want you to know, is that if I ask you about the functions of alpha, beta, and gamma interferon, the most basic is what appears on page 442. And that is that alpha and beta is involved in antiviral activity. Okay? It's the one where, we taught that a virus infects the host cell, and that cell can't save itself, but it causes the production of interferon that goes out and can protect other not yet infected cells. That's mostly an alpha and beta job. That's mostly what alpha and beta does. Gamma doesn't. Okay? What gamma does is has an effect on phagocytes and enhances their activity. So macrophages and neutrophils. It's not all gamma does, but gamma doesn't have that same kind of antiviral effect that alpha and beta do. So if I ask, that's the answer I want you to give. Okay? We're good? So I'm not going to ask you the more complex, under the radar kinds of things that alpha and beta interferon can do. Okay. Vaccines, we talked about vaccines somewhat, and now we're going to go back to the beginnings of this idea of vaccines. We kind of talked about this too when we talked about cows and cowpox and cow farmers having some immunity or protection when smallpox came into the area and was just wiping out families and communities. Those with experience and up close and personal contact with cows who had cowpox had some protection because their bodies had produced antibodies to cowpox that had some cross-reactivity with smallpox, giving them some protection. So, vocabulary lesson, variolation is was our first real dabbling into the vaccination process. And you would take some smallpox or some goop, some grunge, some crust from a person who had active smallpox, take it from an infected lesion, and you have the person who wants to be protected to sniff it. So they sniffed in some of this antigen, and hopefully, not enough of the antigen to make them have a full-blown case of smallpox and then die from it, but just enough to let its body know that there's an invader here and we need to make antibody against it. And it really did work. or if it didn't kill you, you were immune when smallpox came to your area. So it's kind of a, you think people are worried about vaccine safety nowadays. People were running to this thing saying, like, everybody, almost 100% of people, unless they have this protection. If when smallpox comes, and it's coming, because it's just bowling over Europe, at least if we do this thing, we have a shot at surviving it. So between 1 and 2% of those who got the variolation, did the sniff, would die as a result of variolation. But 30%, if they didn't and they came in contact with the smallpox organism naturally, or people started to get it, they made contact, and so looked like pretty good odds back then. This again talks about some of the early history of smallpox, and even royalty got involved, because everybody was kind of scared of this idea of having 30% of your family being wiped out when smallpox came to the area. So it's interesting. You can go back and have a look. And it's interesting, you might see on Facebook about vaccines and how Africans really brought this idea and introduced this idea to the North America, to America. And so it was like 1700s-ish when this thing really started to take hold. Of course, never risk-free. 1 to 2% were going to die of it. The people who were protected would get a small, like, febrile illness, might have gotten some lesions on their skin, but would have a protection when an epidemic really came to their area. Vaccination, inoculation of small cowpox into the skin. This was generous, a little bit later than the snipping the lesions, and he recognized this protection that people who had contact with cows had. And now we have this term called herd immunity, which you may have heard when you took undergraduate microbiology. And what it is this is what people who are against vaccinating their children, this is what they're depending on to keep their children safe. The herd will keep their children safe. So if all of us decided, okay, we're going to bite the bullet and we're going to get vaccinated against measles, just say measles. Seems to be the one I've been choosing lately. We'll continue with that theme. We're vaccinated with measles, so we have antibody in our circulation against measles, and we have memory cells in our bone marrow, plasma cells in our bone marrow, just kind of squeezing out little bits of it. We have memory cells in our lymphoid tissue, so we're good. If a new student comes into the classroom whose mother said, no vaccines for my kid, no way, it's too traumatic to the immune system, it's a stress, it's an unnatural stress to their bodies, it's a political conspiracy to control our children and their health, and only a parent should be able to make this decision, and you can't make me make this decision, and I'm gonna not do this, okay? So their kid, this new student does not have any antibody, so he comes in and he takes that seat right there. And so now somebody comes in with this awful rash and this fever, and it looks like they just want to lay down and die, and they cough and it sneezes all around this place, and just spreading active measles virus in the classroom. Well, if he sneezes right here, all over the front row's got an antibody, the antibody's going to start neutralizing the virus and all that stuff. And he's neutralizing virus, you're neutralizing virus, and you're neutralizing virus. And so the herd is keeping the virus from ever getting to the guy over there in the middle seat. So that's what the herd immunity idea is. If most of the people in the community are vaccinated, those few who aren't vaccinated will benefit from all the immunity that's around them. Because you're not going to get the infection, and you're not going to get it, and nobody else in this room is going to get the infection. The problem is, we are the plastic bubble for that person, but that person is going to get in an elevator. That person is going to be served in a restaurant. That person is going to have contact outside of the herd. So that person is susceptible to the infection. And then once that person is infected, because they don't have any antibody and they're outside the herd, they become a source of the infection for everybody else who is unvaccinated. Either other people whose mother said, no, never, not my kid, or somebody who has cancer, leukemia, has on chemotherapeutic drugs that knocks out their immune system, little bitty babies who are too young to be vaccinated, real old people whose immunity has waned. So they serve as a source or a nidus for infection to move through a community. And so we go through having a low endemic level of measles to having a big blip, and that's happening across the country, little pockets of epidemics because of this very thing. So that's what the herd is. When there are more people who are immune in a population, it decreases the likelihood that a susceptible person is going to get that infection. Okay. Contact immunity is an interesting way of becoming protected without getting a shot. And this kind of immunity is the kind that used to be conferred or potentially conferred when we used the oral polio vaccine. Anybody remember the old oral polio vaccine, the drops? And that used to be my favorite. I was happy to go get that shot. Go get your boosters. It's almost time to, school's going to start. Do I have to get a shot, or can I just get the sugar? Can I just get the sweet stuff? When it was the polio booster time, I was all for it. Yeah, let's go to the clinic because nothing hurt. But the reason that it's such a good vaccine is because the virus is attenuated. It's not dead. We're not using little bits of the virus for antigen. It's the actual virus that can actually take over the machinery of the host cells to make more and more virus. So you give a little bit of virus in the vaccine, a little bit of antigen, but you get more and more and more antigen once it's in the GI tract. So that's why it serves as a great vaccine. Problem maybe it's not a problem. Let's look at it as a non-problem first. Let's say that we have one of these people who say, no, I don't even want my kid to get this oral sweet drops because it is also an unnatural contamination to the purity of my child's body, and I'm not having it. With this kind of vaccine, if that individual decided they were going to be an au pair, they were going to be a nanny or a babysitter, and they were taking care of a kid who, recently had their vaccines, and they were in charge of cleaning, changing Pampers and all those kinds of things. The GI tract of the baby who's been vaccinated now has live, attenuated virus in their poop. So with diaper changes, the unvaccinated person is coming into contact with virus. That virus can come, fecal, oral, root, whatever. It got in that the unimmunized person's GI tract, starts to take over the machinery in the host cells, starts to make virus, starts to make antigen, and that person is making protective IgG antibody, even though they never went and paid for the vaccine. So they got contact immunity. They came into contact with the virus and became immune that way. The reason that we don't use this vaccine in the United States anymore is because people who, the epidemiologists who, do this work and collect the data and draw the charts and the pie charts and the graphs and all that, determine that the risk outweighs the benefit. Why? Because we have so many people in this first world country who are walking around with immune systems that don't work very well. And in those people, like people with HIV/AIDS, they look good, they're healthy, they're taking their medicines, but their immune systems, not so great. We have people who are being older and older and older. The population is getting older. Their immune systems aren't so great. We have people who are living after bone marrow transplants and kidney transplants, and all these people in our community are immunocompromised. Immunocompromised people can come into contact with a baby, a person who's shedding virus, who's shedding this attenuated virus, and an attenuated virus in a person with a normal GI tract will never get polio. But if your immune system doesn't work, and we have seen this, the numbers of cases of paralytic polio in the United States started to have a little uptick. And the epidemiologist says, oh, no, we can't do this. We gotta get people immunized another way. We can't have all these babies running around with attenuated virus and coming into contact with people who already got enough trouble. They don't need polio, paralytic polio on top of it. So now, all polio vaccines given in the United States is an injection. Okay? It's no longer, the oral polio vaccine is no longer an option in the United States. Still a great vaccine, still a cheap vaccine, still an easy to give vaccine, so it's in mega use in third world countries because you don't need alcohol wipes, you don't need needles, you don't need any of that stuff. You just give them little drops and send them on their way. So the United States no longer gives OPV or oral polio vaccine. We give IPV, it's an injection. Okay. This is what happens during vaccination. We put antigen under the skin, and the antigen is recognized as non-self, and the body does all the things that you know and starts to make antibodies. So if in the third box over there, the host comes into contact with a real or wild type virus or organism. There are going to be antibodies in the system, in the circulation, plasma cells in the bone marrow, blah, 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 in the lymphoid tissue, and there's going to be neutralization of the disease organism so we don't get sick. So that's how vaccines work in the simplest form. These are not all, but a good idea of some of the preventable vaccines that are used in the U.S. Most of us never have vaccination against rabies, but the rest of them, yes. Now, what are vaccines made out of? Some are killed host cells, not a whole lot of those. Some of them are inactivated or attenuated or pieces of viruses. So these viruses can't replicate. They can't get inside cells. They can't make more. They don't become better antigens inside the host because they're totally inactivated. We're just injecting them so that they can act as antigen. So it's a real virus enters the host, they're protected. Live, attenuated cells or viruses that we talked about, the polio. Antigenic molecules, so you take the virus or the vaccine and you break it up into little pieces, and the vaccine makers choose the most antigenic epitopes and purify those and create vaccines out of those. And we have genetically engineered microbes. Some of the hepatitis vaccines are those. So all different kinds of ways of making vaccines. Let's see what the movie says.
There are a variety of ways to produce vaccines. Traditionally, vaccines have consisted of killed or inactivated pathogens. A second traditional approach is to use an attenuated strain of the pathogen. During attenuation, the pathogen loses its virulence, but retains many of its antigens and is therefore able to elicit an immune response. Subunit or acellular vaccines consist of selected components from the pathogen, such as a surface structure, an internal macromolecule, or the flagellum. Recombinant vaccines are produced by cloning the gene for a structure, such as a surface protein from a pathogen, into a host microorganism. For example, the gene for a surface protein of the hepatitis B virus has been cloned into a yeast cell. The yeast produces the virus surface protein, which can be purified from the growth medium of the yeast and used as a vaccine.
So, what makes a really good vaccine? The best vaccine would be one that you gotta inject somebody. It would be great if that was the worst thing that happened to them, that they got COVID. Okay? And they cried for a minute, but it was over. Everything else was good. So a good vaccine would have very little in the way of adverse effects. So you have very little to say to the parent that there's not going to be any soreness, there's not going to be redness, there's not going to be, the kid's going to be like nothing ever happened. Usually there's something. A low-grade fever, sometimes not so low-grade fever, tenderness in the area, but those are things that, hey, you don't feel that great for a day or two. But the upside is your body's going to have protection against this organism. So low level of adverse effects is a good thing in a vaccine. A vaccine, if you're gonna get poked, at least you should be able to say, now you just don't have to worry. Because if the child contracts this, comes into contact with the wild form of this pathogen, we don't have to worry because they have antibody against it and they're not going to die from the horrible disease that can be caused by the pathogen. It should stimulate both B cell and a T cell mediated response, and we like immune responses that involve T cells. Why?
Memory.
Yeah, because we got memory. So we can feel really pretty good that if we do become exposed, we got memory cells, and really we got antibody circulating right now because of the active nature of the body's response to the shot. But if we come into contact, we're going to get even more antibody response. So yeah, we want the T cells involved. Should have long-term effects. Yeah, that's the memory thing. And then we talked about over a long period of time, there can be waning immunity or decreasing titers of or concentration of the antibody in the system. We want ones that have long-term effects. So then we get to the next bullet point. We don't need very many boosters. A booster that you need only every 10 years is awesome. A booster, one that you never need a booster for is even better. There are not a whole lot of those. Should be inexpensive, so it has it's not prohibitive for people who have this way of protecting people, but it's too expensive for them to get. That's no good. You may as well not have a vaccine. A long shelf life is good, particularly if you are trying to immunize a population that's in a remote place. People have to come long distances to get. You can't just say, okay, I'm ordering it. It's going to be in my office on Wednesday. Be in my office on Wednesday because it's not going to be any good on Friday. Well, that's no good. So you want ones that are going to be okay. It's going to have a long shelf life, and it would be really good if you didn't have to handle it in a special way. Like it must be kept between -2 degrees and -30 degrees, or it's no good. Long shelf life and easy management requirements to keep the vaccine active and easy to administer, not only for the provider, but for the person who's going to get it. I hate shots myself, but anyway. So these are some of the features that make for a good vaccine. And we said sometimes we use kill whole organisms or kill whole viruses. Whatever we do to kill it doesn't destroy the surface or the antigenicity. Notice the second bullet point makes this different from the live attenuated ones that we talked about. We said live attenuated ones, you can use a small dose because the antigen is going to make more of itself once it's in the body. So these that are dead, you have to give a larger dose, and you may need more boosters than you would if the antigen just kept making more of itself over time inside the host. These are live attenuated ones. We take away its ability to produce infection. Well, not really. We take away its ability to produce disease. Okay, we want it to produce infection, or we want it to get inside the body and make more of itself. That's what we mean by the infection part. So it produces infection, but it doesn't produce disease. That just means it's inside the body and it continues to make more antigen. Long-lasting protection, we told you why. Fewer doses and fewer boosters, we already talked about that. Disadvantages is the storage thing. These are live organisms, so you gotta take care of them like they're alive. Okay? You have to keep them at a certain temperature. If your refrigerator or freezer breaks down and it's warm in your freezer, you just don't. They're dead and they're not gonna work as a live attenuated virus will. Problem as a disadvantage, we talked about these having contact with people who do not have an intact immune system, and having even the attenuated form of the virus or the bacterium able to cause disease when the immune system can't combat or keep it at bay. And a real big problem that you want to avoid is it mutating backwards to a virulent strain. It doesn't happen very often. Antigenic molecules, like our little Fluky said, we can take pieces of the bacterium or pieces of the parasite or the virus and purify those and use them for the vaccine. Use pieces of capsules, surface proteins, and we said that the hepatitis B is actually a genetically produced surface protein, and make it inside of a yeast cell. So you don't necessarily have to break up the thing and take the surface protein. There we do have technology, genetic technology, to make the proteins that we want inside another organism, and then purify it and use it in that way. Toxins. Now, the diphtheria and tetanus, the organism itself is not what makes you sick and kills you. It's the toxin that it emits. So the vaccine against diphtheria and tetanus is actually against the toxin. So it doesn't stop you from being infected with the organism, right? You can still step on a rusty nail. You can still have the organism get inside of you and start to infect your foot, let's say. But once they grow, it's not the organism that's the problem, it's the toxin that they produce. So that once they start to produce this toxin, you have antibody that's going to clamp down on it, and it's never going to make you sick. You just have to take care of the wound and clean it, and antibiotics and whatever. But the organism itself is not what's going to be the death of you. And we already talked about synthesizing antigens and genetically modifying it. And this is just one of those pictures that I like, where everything is kind of in one place. Everything we talked about already. We can take a whole organism, kill it, and inject that, and we know the pros and cons of those. We have live cells or viruses, and we treat them with, oftentimes it's some sort of a chemical compound that takes its virulence away, but doesn't take the ability to replicate away. So you administer those, and they continue to multiply, so it's a great way to give a vaccine, but we also understand that there can be potential problems with those. Subunit vaccines, we take it, we break it apart, we take the pieces that we want, and we purify those, and we can use those as antigens in our vaccines. And we can do the fancy stuff, genetically produce a protein that's very antigenic, and then purify that protein that comes in this situation from a yeast and use that as a vaccine. So that's pretty much all the different forms of vaccines that we have. And been working for years and years and years and years on trying to come up with an AIDS or HIV vaccine that works. Been working on herpes and problems with the big problem with the AIDS and herpes is mutation. Once you get a vaccine made that's going to elicit an immune response and make IgG, the organism has mutated and that antigen doesn't exist anymore. Another problem with the HIV virus is that it can mutate inside the person. So the organism that infected them initially is different from the organism that's in them 2 years down the line. So they can change their antigen during the process of infecting the individual. So all kinds of stuff going on, trying to make newer and better vaccines. It'd be wonderful to be able to get a malaria vaccine that works. Again, they're working on that for years and years and years. There are usually several that are in testing at any one time. We just don't have one that's ready for prime time yet. Influenza, we'll talk when we get to the virology section, is one of those viruses that we know they have to make a new batch every year because of mutation, antigenic drift and shift, which we'll talk about, because the antigens on the surface of these RNA viruses are the ones that mutate very, very, very regularly. Because, and the reason for that you'll understand down the line, but a little preview is that DNA virus has this enzyme, DNA viruses have this enzyme called DNA polymerase, and DNA polymerase has the feature of being able to fix problems. There's a problem in the DNA, it'll cut out bad sections and fix them. There's no such thing as an RNA polymerase, so problems that occur in the genetic material in the RNA can't be fixed. So that's why we get new antigens on the surface of RNA viruses very, very frequently. And the problem with influenza and why we have to have a new batch of antigens every year for our RNA virus. This talks about the same kind of thing as the hepatitis and how we go about making vaccines genetically with plasmids and DNA. How do we give vaccines? We know most of them are injectable. Some of them are oral. I wish more of them are oral. At least we have some nasal ones. I guess that's the next best thing to an oral one because it doesn't hurt. So we have sometimes. Has anybody ever had a nasal influenza vaccine? One, two, a couple of you. I haven't. I do get it. Most years I get it. I didn't get it last year, and it turned out to be a good year not to get it because it wasn't a very effective one. But I just didn't get around to it. Yeah, probably as we move forward in time, there'll be more and more of those that they can find ways to make them antigenic, inhale them. And it will have to be, you won't be able to give a malaria virus nasally because it doesn't have there's no part of the life cycle of that organism that's respiratory. So usually the nasal administration ones are ones that you're going to come into contact with the wild form of the organism by breathing it in. Okay, adjuvants. Some vaccines require adjuvants to enhance immunogenicity, and prolong retention of the antigen. This should remind you of something that we talked about before. Having to stick an antigen on something so that the immune system has an easier time of seeing it, making it more antigenic. What did we talk about? There was something like that.
Haptens?
Huh?
Haptens?
Haptens, yes, very good. Haptens are too tiny for the immune system to see. You hook it onto a protein and then it says, yeah, that's non-self, and we should probably make antibody against that. So, same thing happens with some vaccines. We make these little bitty pieces of antigen, but some of them are too small to have a good response in the host. So we have to hook it onto something, and usually it's a protein. You know, you gotta have more benefit than this, and these are the typical side effects: injection site, tenderness, soreness, swelling, fever. Occasionally people are allergic to some of the components. Anybody know of a particular vaccine that if you're allergic to, you probably shouldn't get this vaccine?
Eggs.
Huh?
Eggs.
Eggs what? If you're allergic to eggs, you shouldn't get the MMR.
Oh, not the flu shot?
No.
They always ask.
That might be a chicken one.
Huh?
Nothing, I was saying what Bob said.
What did you say? What did Tom say?
They always ask if you're allergic to eggs when you go to get the flu shot.
Has anybody ever said yes?
I think that's because, did we say something about live virus for some flu shots and others aren't live virus, so they grow up in eggs?
Some of them are virus as an egg. What? No, that's typically not one. If you have like anaphylactic response to eggs, maybe, and then maybe some batches of flu vaccine will come with a special disclaimer. Find out if patients are allergic to eggs, but typically the vaccine that is across the board prepared in eggs is the MMR one. And usually, if you say yes, that's still not reason enough for you not to get it. They may do a small skin test, put a little bit beneath your skin to see if you respond. Bring you back if you do okay. If you get this big giant necrotic thing, no, you're not going to get that vaccine. But usually, most people can get the vaccine, but there is that precaution. You need to find out, and you need to test. Okay, perfect. All right, let's take our break. And I'm not really sure what happened to our folders, but I'm going to remake the folders. Professor Pat showed me that there is a place on Blackboard where they took all of our folders and dumped them out into a into, another place, and I'll have to make new folders and drag them back in for you, so it's easier for you to find our stuff. But I posted 16 just right in our course contact folder, so we can find that for now.
Jotted